And I'm going to start it out by showing this slide from a very well-known review by Mike Yassa and Craig Stark in Trends in Neuroscience about pattern separation in the hippocampus. Mike and Craig wrote this when Mike was at Hopkins during his first faculty position, and he and I used to talk a lot and collaborate. And we were talking about this figure. On the left here is this very well-known type of figure about showing pattern separation and completion. The idea of when you change the input into a system, how is the output affected? The idea is if the output is affected less than the input, you can call that pattern completion. If the output is great, change in output is greater than the change in the input, pattern separation. And the idea that dentate gyrus of classic models is doing pattern separation. It very rapidly changes outputs for minor changes in inputs. CA3 with attractor dynamics would start out doing some pattern completion until the output was so the input was so different, it would have an output that was more different. And CA1, more linear, would be an input-output function. And that's very classic ideas, from the work of starting with David Marr's work, people like Bruce McNaughton and Lynn Nadell, Evan Rolls, Alessandro Treves, and others, Jay McClellan. This is all due to computational modeling to put these ideas out there about these different regions. And then Mike was also showing work he did with Craig and in human imaging that when people were seeing repeats of stimuli or stimuli that were very similar to each other or not very similar or completely new, would show a linear relationship in CA1 in terms of beta coefficients in the bold imaging. And their dentate CA3 imaging, they couldn't distinguish the CA3 from the dentate. It would look more like this, much more like the dentate would than the CA3. And the way they thought about this, well, maybe it's they're combining CA3 and dentate together. So you add the red curve to the green, you get something that looks kind of like this, and that's what it looks like. And Mike and I talked about this a lot, and it really puzzled me because this kind of assumes that the dentate and CA3 would have equal weight to the bold signal. But at the time, people thought, and now continue to think, that the dentate gyrus is a very sparse encoding system. Very few dentate cells fire. So it really never made sense to me. If CA3 is supposed to be doing more pattern completion and maybe more sigmoidal responses, why this seems to be more dominated by what you might expect the dentate to do. So that was just a conundrum. The data are the data and how you think about it. But hopefully, I think now we may have actually an explanation for what's going on here. So I'm going to talk about that in this talk. The outline is going to be such. We're going to talk about pattern separation and completion on the CA3 transverse axis. The second part talk about granule cells and the dentate gyrus, Mossy cells, and CA3 C-pyramidal cells constituting a functional unit for pattern separation working together. And finally, implications for memory impairments in aging. So the first part of the talk, is there evidence for pattern separation of the dentate gyrus and pattern completion or attractor dynamics in CA3? When I say pattern completion, I'm talking about not just classical pattern completion, taking a partial input and reconstructing the full output, but even things like an attractor network should be able to do, having a corrupted input and also retrieving the original input, or doing error correction, things like that. All the same attractor networks should be able to do all those things. I'm just going to put them all under the rubric of pattern completion. So here are the experiments that we've been doing for many years now, where we have a rat running around a circular track, and there are salient local textures on the track. And then there are the standard visual cues on the curtains, circular ring of curtains surrounding the apparatus. And from an overhead schematic view, here's the rat that would run around this circular track. Here's the black curtain with the different cues. And after the rat has many days of experience running around in this standard environment where the local and global cues are in this configuration, we'll do experiments like recording the cells in the standard session. And then when we take the rat out, we rotate the track counterclockwise, we rotate the cues on the curtain clockwise, and we give a cue mismatch between a local framework and a global framework, and see how that affects the hippocampal rat maps. Here are some examples of the types of individual responses a place cell will do. So here's one example. We do a standard session. We do one mismatch session with a 90-degree mismatch. The track is rotated 45 degrees one way, the global cues 45 degrees the other way to give a total mismatch of 90 degrees. Return back to a standard 2 session like this, and now here's another session, 135 degree mismatch. So this is the standard sequence of the experiment. And we see some cells will rotate their place fields with the clockwise to go with the distal landmark. So here's a cell that had a place field at this southeastern location, and it rotated about 45, 50 degrees in the clockwise direction, following the cues on the wall, on the curtain. Here's another cell that also rotated in the clockwise direction. This is a 180 degree mismatch. So the cell didn't rotate its field exactly with the cues, but it went in the direction of those cues. Other cells rotated counterclockwise. Here's a cell that rotated in the counterclockwise direction, its place field. Here's another cell that was fired here. It rotated its place field in the counterclockwise direction. And then other cells did a remapping. They would either have a field in the standard session and would completely shut off in the mismatch session. Here, the cell would return again in the standard, and then in the mismatch session, it just got really confused about where to fire. And similarly, we had other cells that were silent in the standard and would get a place field in the mismatch. So we got a whole mixture of different cells. Some cells rotate with one set of cues. Some cells rotate with the other set of cues. Some cells develop a new place field in the mismatch session. Some lose their field. And all the different areas I'm going to talk about and all the types of animals all have examples of these cells. But there are different proportions of these cells and different measures of coherence of what the ensemble or the population does. And that's what we'll be looking at here to make our conclusions. So the first question is, this is work that was done by Josh Nunabel, a grad student in the lab. He's now at the University of Delaware as an assistant professor. The question is, when looking at dentate gyrus cells versus CA3 cells, do we see a difference in how these populations of cells respond to this cue mismatch? We're looking at these population correlation plots to look at that question. The first thing we'll look at is a standard 1 session versus a standard 2 session. So these are 2 sessions that had the same standard configuration of cues. And what we do was to take, for each degree bin on the track, we would look at all the firing of the cells, of the population of cells at that 1 degree of bin, that 1 degree bin, and then we would correlate that vector of all the cells firing at that location against all the locations standard 2. And here, all the locations here are correlated with all the locations of standard 2. The upshot is that this band of high correlation on the 45 just means that the maps are very stable between standard 1 and standard 2. Cells that fire at this location in standard 1 fire at the same location in standard 2. Cells that fire at this location in standard 1 fire at the same location in standard 2, and so forth. So this is just telling us it's a coherent map. The map is reproduced faithfully between 2 experiences of the standard environment. And you can see both CA3 and dentate gyrus had a similar band of high correlation. Dentate wasn't quite as nice as CA3, but clearly there's a correlation band there. Now what happens when we look at standard versus mismatch? Well, what Josh did was replicating work we earlier published from Ina Lee in the lab, where here in a 90-degree mismatch session, we still see a band of correlation along this diagonal here. It's shifted off the axis, the main diagonal. That means the map was actually controlled by the local cues on the track, also what we have seen many times before in CA3. But you can still clearly see this band of correlation showing there's a fair amount of coherence in how the map rotates in the standard mismatch session when there's a cue mismatch. Not all the cells rotate as a coherent population, but enough do that you can still see this band of correlation. However, that band of correlation is missing the dentate gyrus. So the dentate gyrus responds to this cue mismatch by more or less completely jumbling up its place fields. A lot of remapping, cells rotate every which way. There's really no coherent response to the manipulation. And that's what you would then expect. You change the input to the system, dentate gyrus dramatically changes its output, whereas CA3 is changing its output much less. So there is more pattern completion, separation going on in dentate, more pattern completion in CA3. So suggestive that there is this difference between the dentate being more involved in pattern separation, as classic models suggested, CA3 with its recurrent collateral system being more involved in pattern completion. Now, that all sounds fine, and this is old stuff. We published this a number of years ago, and it's consistent with the classic models, but these models are all based on the idea that the dentate gyrus is a unitary structure, and that CA3 is a unitary structure, and CA1 is a unitary structure. And we know, of course, along the proximal distal axis, the anterior posterior axis in primates, the so-called longitudinal axis, that's not true. There are a difference in function along that axis, but there's also a difference in function along this so-called transverse axis here. And that matches up with a lot of the anatomy. So for example, we know that lateral entorhinal cortex layer 3 projects to the most distal part of CA1, whereas the medial entorhinal cortex layer 3 projects to proximal CA1. Layers 2 of NEC and LEC project to both the dentate and CA3. But it turns out actually that the projections to CA3 is much greater out here than here. The reason is that the entorhinal axons come in around here, and they only extend about this far. So if I'm a CA3 cell out the far proximal, I can't stretch my dendrites out far enough to get entorhinal input. So that's one difference, that proximal CA3 gets less direct entorhinal input compared to distal CA3. We also know in the Mossy fiber pathway from dentate to CA3 that both the upper and lower blades project to proximal CA3, whereas distal CA3 primarily gets input from the upper blade. We know that there's differences in projection. Distal CA3 projects primarily to proximal CA1. Proximal CA3 projects primarily to distal CA1. So this topography of projection mirrors symmetrically around the CA3 to CA1 transition. And of course, in the current laterals of CA3 are more dense in the distal part of CA3 compared to proximal CA3. So there's a lot of complex anatomy that goes on in the hippocampal circuit, the so-called trisynaptic loop, that really needs to be taken into account. So based on all this anatomy, we wanted to look at, well, are there differences along the proximal distal CA3 axis in terms of pattern separation completion? The hypothesis was that CA3 with a higher density of recurrent collaterals would show more pattern completion, more attractor-like dynamics compared to proximal CA3, which has a greater input from dentate gyrus, and maybe it would show less pattern completion, more of a pattern separation. So Hee-Kyung Lee and Chung Wang teamed up to do this experiment where they recorded along the proximal distal axis of CA3 all the dots of the different recording sites over 5 or 6 animals, and did similar correlation matrices like I showed you earlier. Proximal CA3 recordings, we had a 45-degree mismatch, a 90-degree mismatch, 135-degree mismatch, 180-degree mismatch. And what I'm showing you here for proximal CA3, intermediate CA3, and distal CA3 are all the standard versus standard correlations. And what you can see very clearly is they all have these nice, strong bands of high correlation along the main diagonal, showing that in all 3 regions, there is a nice stable map between 2 visits to the same environment. But what happens when you look at the standard versus mismatch sessions? If you concentrate first on column 4 and 6, intermediate CA3 and distal CA3, you can see, again, the nice bands of high correlation that we had shown previously, both in Ina Lee's original experiment CA3 and Josh Nudabel's replication of that part. And we also see is that the high correlation bands creep downwards from the main diagonal as the mismatch angle between local and global cues becomes greater, showing again that the CA3 map is controlled primarily by the local cues on the track, not the cues on the wall, and the representation gets a little worse and worse as the mismatch gets greater and greater, but it still maintains a very nice high degree of correlation, a coherent map. Now you contrast that with proximal CA3, and the correlation structure mostly breaks down. This looks very much like what Josh had shown in his dentate gyrus data. So there's a difference between proximal CA3 into intermediate to distal, that the proximal CA3, its response in this double rotation manipulation looks much more like what we found in the dentate gyrus, and the pattern completion type of phenomena, the putative attractor networks, is more in the distal and intermediate part of CA3. So corresponding with the anatomy, we do see this functional differentiation along the transverse axis of CA3. If you're interested, Sachin Deshmukh, who was a postdoc in the lab, has a paper on bioRxiv now looking at proximal distal CA1 in this same double rotation experiment. And if anyone's interested, You can go to bioRxiv and look up Sachin's name and find out what's going on there, but I don't have time to talk about that today. Okay, so let's get back to what we talked about initially about the human work that Yassa and Stark did. Now, in the rat, proximal CA3 or the classic CA3C is the part of CA3 that's enclosed within the blades of the dentate. CA3B is the part from there to about where the bend of CA3 ends, and this curved part of CA3 is CA3A, the distal CA3. And it appears to be the case, though, that in humans, there's a lot more proximal CA3 than distal CA3. And that was suggested in this paper by Clifford Saper's group, where now here's a primate, a monkey hippocampus, and you can see here's a subiculum CA1, CA3 region, and all these cell bodies are in here, enclosed within the blades of the dentate. What this paper and its companion paper from the Saper group argues is that this is all CA3C. And if all CA3, if this is true, and I'm not an expert in human anatomy, so if anyone here knows more, I'd love to hear about a discussion. But if it's true, it suggests that there's a lot more CA3C in the primate brain compared to the rodent brain. And this made, and according to Saper here, they argue that these changes mirror a phylogenetic growth of the CA3 pyramidal neurons, subfield CA3, into the hilus. The growth of this subfield may reflect a growing role of the projection from the granule cells to the subfield CA3C, and from there to field CA1, the primate hippocampus. So if the bold signal in the imaging is combined together, dentate and CA3C, well, that explains, I think, why the bold response is dominated by the pattern separation types of functions, because proximal CA3 appears to be working as a computational functional unit with the dentate gyrus for pattern separation. And this part of CA3, which is relatively smaller in the primate brain, is what's involved in pattern completion. Now that has some interesting implications. If proximal CA3 is doing primarily pattern separation with the dentate, it sends a pattern separated output to the part of CA1 that is connected to the lateral entorhinal cortex stream. It's as if this bidirectional connectivity between lateral entorhinal cortex and distal CA1 wants to get pattern separated input from the dentate gyrus, whereas this part of CA1, which is bidirectionally connected with the NEC, wants to get input that is biased towards pattern completion. So it suggests that we talk often about pattern separation, pattern completion being complementary, competing processes. This model here suggests actually we can have our cake and eat it too. This part of CA3 is doing pattern separation. It's separating inputs, sending that information to the part of CA1 that is somehow more involved with whatever processing LEC is doing, whereas this part of CA3 is biased towards pattern completion, sends this output primarily to the part of CA1 that's bidirectionally connected with MEC for its processing. So whatever this circuit's doing wants input from CA3 that's biased towards completion. Whatever LEC is doing in its bidirectional connectivity with this part of CA1, what's output from CA3 that's doing pattern separation. Okay, so I'm going to finish up now with a little bit about how this may be related to memory impairments in aging. In the Long-Evans rat model, the rats break down, aged rats break down into two categories. This is the work that's been done for decades now by Michaela Gallagher, with whom we're collaborating on these studies. When you have a learning index score in the Morris water maze, a group of old animals have the same learning index as young animals, whereas another group of old animals have impairments. They're worse than the age impaired and worse than young animals. That suggests that the AU rats perform at levels similar to young, but they show other brain changes, suggesting that they can compensate for brain aging better than AI rats. So what, again, Heek Young recorded, a monumental work here, recorded from 7 AU rats, 7 AI rats, 5 young rats, a tremendous amount of work to get data sets in the same double rotation. Young animals, this is just an example of some of the place fields. They look pretty much normal, but when you quantify them, what you find is that in young animals, in proximal CA3, AI animals have less spatial information than young or AU animals. By the time you get to distal CA3, there's no difference between them. If you just look at the young animals, the first bar here, the information gets lower and lower in young animals from proximal to distal, which is what she had seen in her earlier work. But then in AI animals, it's flat. So AI animals lose this gradient in spatial information from proximal to distal. And that's mirrored, I won't go into it, but that's mirrored in the place field size here in the opposite direction, as you would expect. Bigger place fields have less information. So that's one point. Now, the Gallagher lab has also been talking years about how hyperactivity in the hippocampus may be correlated with learning deficits. So Young looked at in the relative distance on the proximal distal axis in terms of hyperactivity in this log scale here with some linear mixed effects models that we use with a statistician, Scott Ziegler, and his student Zitong Wang. We find that in the proximal CA3, the young and the AU animals, the AI and AU animals are hyperactive in proximal CA3 compared to young, but as you move towards the distal axis, that difference goes away, and maybe the trend is even reversed. It's not quite clear. Notice these shades here are 95% confidence intervals, not error bars. So these are clearly very highly significantly different in these models. So the proximal CA3 appears to be where this hyperactivity occurs, not so much so in distal CA3. And finally, just to quickly remind you the place field properties, we get some cells that rotate, some that remap. This is an indication we think of more cells doing what we think is more like pattern completion. This is the remapping, the pattern separation. What Young sees is in proximal CA3, young animals show the classic result, which are more remapping than rotating, which goes to less of a difference in distal CA3. So you get, this is the pattern separation going on in proximal, more pattern completion in CA3. I see the chicken, I'm about to finish. Whereas in AI, that's different. In AI animals, it's the reverse. You get a lot more rotation than remapping in proximal CA3. And by the time you get to distal, very little difference. So it just shows that it appears to be this proximal CA3 that is where the big differences are. So to conclude, this functional heterogeneity around the CA3 transverse axis in terms of pattern separation and completion. Proximal CA3 acts as a unit with dentate. Evolution may have selected an increase in the dentate CA3 circuit in primates, enhancing the pattern separation function of the hippocampus. And age-related changes in CA3 seem to be most prevalent in proximal CA3 compared to distal CA3. I'll just finish up. I mentioned everybody here. A special shout out to Heek Young, who's on the job market, did fantastic work, and anybody who's looking for a great person to do aging research, she's your person. All right, sorry for going over.